Thanks so much, Harold. And so let me just continue here. So tonight I'm going to talk about paradigms of cancer. And really, the idea is to look at the question of what is cancer and how does it develop? It's a question, I think, that we don't think enough about in terms of what this disease actually is. And that's the thread that's going to run through tonight. Uh, how do I? Oh, here we go. So cancer is actually a very old disease. So it's been mentioned even in the Edwin Smith papyrus, which was from the 17th century BC. And even back then, there was cancer. So it's not a new disease. But what's interesting about this disease, it's been sort of growing since that time. So it's relatively uncommon in the old times. And part of it is because we're getting older, but it seems to be getting more common anyway. And the question is why? Most other diseases that we deal with get better over time. We have more better treatments, we understand it better, but this is a disease that we just don't understand. And that's one of the reasons why we probably don't treat it as well as we could. If you look back at sort of the major causes of death, this is from the New England Journal of Medicine, and it looks at the sort of number of deaths in 1900 versus 2010. And what you can see is that most of the diseases in the 1900 have sort of gone away. So, not very many people die of tuberculosis and pneumonia and nephropathies, that kind of thing. But cancer is there, but it's getting much bigger. Heart disease, of course, it was there and it's getting much bigger. And both of these are diseases of aging, of course, and that explains some of the reasons, but these still remain sort of the two biggest things that we need to worry about. Heart disease, of course, we know quite a lot about and what we're doing about it and how to treat it. But cancer is still this incredible mystery. That is, what is this disease? And you can see because we don't really understand it, the sort of pace of progress is very slow. So this is 1969, and this is annual mortality. And of course, if you look at heart disease, you can see that it stays relatively stable, but the rate this is the total number of deaths. So because in the United States, there's more people, the annual rate of death is actually going down. But you see that it stays relatively stable, if not going a little bit down. Whereas cancer, in terms of total number of deaths, just keeps going up. And the idea was that, there's all different kinds of cancers. But one of the sort of major things that sort of advanced us in our understanding of cancer was what makes them cancer? And this was a very interesting question. And two researchers had sort of one day they were at a conference and they decided, well, what is it that makes cancer cancer? Because we talk about different cancer. We talk about breast cancer and we talk about colon cancer and so on. But so we know that they're somewhat the same, but different. Breast cancer is not the same as lymphoma. So what is it? So they wrote a paper, around 2000, which is called The Hallmarks of Cancer. And in it, they set aside six sort of major hallmarks of cancer. And it turned out to be one of the most downloaded papers, most cited papers in oncology. So they added two more emerging hallmarks. And these are those here, but they basically fall into sort of four categories when you think about it. And those categories are that they grow. So if you look at cancer cells compared to normal cells, cancer cells grow, they keep growing. That's what they do. And that's one of the big hallmarks. They're immortal. So that is, if you try to grow normal cells in a sort of in vitro state, they don't grow. They reproduce a certain number of times, then they stop. But cancer can just keep going. They move around, so normal cells don't move around. Like your liver cell doesn't just hop on up to the the lung and just to hang out for a while, right? It stays where it is. The cancer cells don't do that; they move around, and of course, that's one of the major problems with cancer. That is its metastasis. And then in 2011, they added this deregulated sort of cellular energetics, which is this Warburg effect, which is a very, very interesting effect that happens in cancer cells. That is, normal cells have an option between sort of anaerobic fermentation, which is generates sort of a little bit of ATP and lactic acid. So one glucose will make two ATP, two lactic acid. But it's a very inefficient way of generating ATP or cellular energy. And if you use oxygen, you can get 36 ATPs, and that's oxidative phosphorylation. So when oxygen is available, almost all our normal cells choose OXPHOS or oxidative phosphorylation because you get so much more energy. When there's no oxygen, like when you're sprinting, then you go to anaerobic metabolism, which is produces lactic acid, you get the burn, and then you stop. However, cancer cells are very strange in that even when there's oxygen, they use this sort of anaerobic fermentation process that generates a lot of lactic acid. And the question is really why? It's not an accident because almost all the cancers that you look at do this. So it's not some big coincidence. So when you think about what is cancer, we've gone through sort of three major paradigm shifts. And the first thing to understand is that the paradigm of cancer has real major implications on how you're going to treat this cancer, because you have to understand what it is to have some sort of rational treatment. So the first sort of modern paradigm of cancer was this sort of growth paradigm. That is, we have cells and they're just growing a lot. And that's the disease. That's the whole thing. What causes this? And some people say, well, we don't know what causes cancer, but that's actually not true. We know a lot about what causes cancer, and those are called our carcinogens. So we know, for example, that tobacco causes cancer. We know that soot causes cancer. So this is one of the very first described carcinogens, actually. And this is a London child sweep. So after the Great Fire of London in the 1600s, they changed the fire code. So they made the chimneys narrower and they had all these bends in it. So you couldn't sort of go up to the top and just drop your cleaning thing down. So instead they sent little boys and girls up the chimney. And of course, many of them died. But the ones who didn't die wound up getting scrotal cancer. And one of the things that they noticed, of course, was that it was always in London, in other places where they didn't have this soot, the children weren't getting scrotal cancer. It turns out that the soot was probably the carcinogen. It got caught in between the sort of the folds and then caused a chronic irritation, which caused scrotal cancer. When, of course, they stopped sending children into hot chimneys, that whole thing just stopped as well. And we know things like asbestos causes cancer, this mesothelioma. We know radiation causes cancer. And this was a picture of the radiation, the radium girls, which was these women who would paint radium onto watches so that they would glow in the dark. Turns out it was very fine work, so that they would actually put the brush into their mouth to dip into the radium. They would ingest a lot of radium and became radioactive. They grew these horrible, horrible cancers. And we know that certain infections cause cancer too. So this is Epstein-Barr virus, and this is Burkitt's lymphoma. So now we're starting to think about sort of what is cancer, what causes cancer. And this was the great growth paradigm, which is that you have these chemical things, you have physical things, you have infectious things, and they all cause the cells to grow. And that's what cancer is. We didn't know how. The mechanism was quite unclear at the time, but we knew it grows. So under this paradigm, how do you treat it? Well, if something grows, then you kill it. That's basically all we did for a while. So surgery, for example, was one of the main things that we did, and we still do, in order to get rid of the cancer, because you just cut it out. You can also burn it out. So you can use radiation to burn these cancer cells, and that's been very successful. And then the mustard gas of World War I led to a whole bunch of innovations in terms of chemotherapy, where you're now going to poison these cancers. And the idea is that you're going to kill the cancer cells slightly faster than you're going to kill the normal cells. So this growth paradigm actually formed the basis of most of modern oncology. And as we learn to sort of use them in better technology, better chemotherapies, better combinations, that produced most of the advances. So this was one of the sort of great advances of the sort of going through the 1950s and 1960s. This is what we went through, this growth paradigm. So it was very successful. But it didn't really answer the question of how this happened, because we knew that these cells were growing and growing and growing, but we didn't know why they were growing. So now, as we try to understand this at sort of a deeper level, we want to try and understand what is it that's taking it from a chemical to excessive growth. And this led to the sort of next great paradigm of cancer, which began with the sort of genetic revolution. That is Gregor Mendel with his P studies, and then Watson and Crick with the isolation of the DNA, the structure of the DNA, led to this genetic paradigm.
Thank you. I'm on tonight. However, I'm not the nurse tonight. It's going to be Seth.
Oh,
Seth.
So this led to sort of the genetic paradigm, which was that, hey, maybe the way that these cells are growing is that they have mutations in the sort of growth genes. So this seemed to fit perfectly, because now you can take all these sort of disparate causes of cancer, and then you can link them into a common mechanism, which was mutations in growth genes. And this is what causes a cell to grow excessively. So this was great. And then they discovered oncogenes, and then they discovered tumor suppressing genes. And this seemed to make a lot of sense. So this sort of coalesced into the somatic mutation theory, which sort of dominated cancer and cancer research over the last 50 years. And the idea with the somatic mutation theory is that somatic means that it's not germline. That is, you're not mutating ovaries and sperm and stuff. You're mutating normal cells like breast cells. Those are somatic cells. The somatic mutation theory said that, well, when you go from a normal cell, you get these sort of multiple sequential genetic mutations. And they're random, because if you look at something like asbestos, it's not a targeted genetic mutation. It's not like you have, it targets one specific gene. Same with tobacco smoke, same with soot, same with any of these things that we knew caused cancer. They were not targeting specific series of genes. They were just random. And the idea was that just by random, you might hit something. So the more times you got these mutations, the more chance you're going to hit one of these critical genes. And then as you get these sort of multiple mutations, it led to cancer, and then cancer would just sort of keep going and proliferate itself, and all of the progeny would be sort of clones of this gene. So this was the sort of somatic mutation theory, and right away they started to see some incredible successes. So there was, of course, the Philadelphia chromosome, which was that they had this abnormal BCR-ABL kinase in chronic myelogenous leukemia. And there was this problem between the chromosome 9 and chromosome 22. You got the Philadelphia chromosome. And therefore, when they understood that this was a genetic condition, they could then say, hey, how are we going to affect this kinase? And the first drug that came out was Gleevec, which was a phenomenal drug. Like it just changed the whole course of this disease called CML, which was relatively rare, but before that, you basically didn't have much hope. After that, you took some pills and boy, you're almost back to normal. So by 2000, the optimism was just, off the charts. So Time was saying there's new ammunition in the war against cancer. These are the bullets. So these are now the new sort of genetic treatments. And the next one, which came shortly after, targeted the HER2 new gene, which was again this. You could look at these cancer cells and say, "Hey, look, they have all these abnormal genes, and how are we going to target it?" And of course, not all the cancers had this, so they actually developed the test and said, "We're only going to treat those people who have that." again, just a phenomenal, like a mind-blowing sort of thing. Instead of just trying to cut, burn, and poison, now we had treatments that were directed against the genetic thing. So the new bullets that they're talking about no longer follow the growth paradigm. We're not trying to find things that kill cells faster. We're trying to actually fix the genetic problem, which was in fact, just unbelievable to somebody who sort of, I grew up around that era. So now we had this paradigm which seemed to fit perfectly, right? You had all these things, you had genetic mutations, you affect it with imatinib, trastuzumab, which now blocks your cells from causing that excessive growth. So by then there was this huge optimism. We're going to cure cancer. That was going to be it. All we had to do was find the cancer, find the gene, and then target it somehow. So for colon cancer, we're going to find this, what the problem is. We're going to find the 1 or 2 or 3 genes that are a problem. We're going to give them the drugs. We're going to fix it. And then we're going to cure cancer. So of course, there's this huge human genome project, which finished around the 2000s. And, we thought, well, once we get that, we're done. We're going to cure the whole thing. It's a done deal. Of course, it didn't. And when the Human Genome Project finished, there's a much more ambitious project called the Cancer Genome Atlas. And this was sort of many times more ambitious. What they did was they took all kinds of tumors. They took 33 different tumors, 10 rare tumors, 11,000 patients. They generated a huge amount of data. They sequenced the entire genome of these cancers, because what they wanted to do, of course, was take these, 1,000 people with breast cancer, and then let's see what the common mutation is, the somatic mutation. And of course, it really just didn't find anything. It was published in 2018, the final Pan-Cancer Atlas. And by that time, nobody even cared about it. You didn't hear about it on the New York Times. You didn't hear about it on Time magazine. It was as if we didn't do anything. And it was a massive program. And the problem wasn't that we didn't find the mutations. The problem was that we found way too many mutations. So this is a catalog, the Cosmic Catalog, this is from 2018. And this is the total number of somatic mutations they found in cancers. So even if you look at just coding mutations, there are close to 6 million coding mutations found in those different types of cancers. And that's not even including all these other ones, 9 million gene expression variants, like just an unbelievable number of mutations. It wasn't 1 or 2 or 3. It was a lot more than that. And that, of course, is a huge problem for the genetic paradigm, because you can't come up with 6 million drugs. It's just not going to happen. And by the sort of 2010s, it was obvious that the somatic mutation theory was just in huge trouble. Because remember that in, say, colorectal cancer, in the 90s, we were talking about these 2-hit hypothesis. That is, you needed 2 genetic mutations. So you affect one oncogene, then you affect another one, maybe a tumor suppressant gene, and then the combination of these 2 hits caused you to get cancer. But there wasn't 1 or 2. In the average colorectal cancer, there was like 70. When breast cancer was like 60, and you can go all the way down. And the problem was that there was mutations everywhere. So if a breast cancer patient A had 60 mutations, the person sitting right next to him, which might have clinically exactly the same type of cancer, would have 60 completely different genetic mutations, which means that you couldn't treat them based on this genetic paradigm at all. It just would not work. You look at this is from Bert Vogelstein in Science in 2013, and you see that, all of them are like this. So colorectal had a lot, lung had a lot, 150. This is sort of average number of mutations per cancer. All of these had, lungs cancer had 160, breast cancer had 30. All of them had multiple ones. Kids, of course, had less. It was much more genetically linked. And there was mutations everywhere. Like, it just it wasn't just that patient A, patient one, and patient two had different mutations. So they would have completely different mutations. But two tumors within the same area also had different mutations, and metastases had different mutations. And even within the primary tumor, different parts of the primary tumor had different mutations. So even if you could figure out this 70 mutations of colon cancer, you wouldn't affect the metastasis, say, or you wouldn't affect the, interpatient problem. So there was just mutations everywhere. And at the same time, some cancers had no mutations at all. So this is a paper in Nature. And when they looked at their number of DNA mutations, yes, many of them had 70 or 80, but 35% had none at all, which is a problem because if cancer is caused by gene mutations, how is that even possible? So the whole problem with genetic heterogeneity went totally against the somatic mutation theory, which was that all of these were supposed to be genetic clones. And a lot of the sort of idea of the genetic paradigm had this sort of denominator problem, which is that they only look at the numerator and not the denominator. So let's give you an example. So if you have 100 professional baseball players and you say, what's common about all these 100 baseball players? And you say, all of them have a liver. Then you say, therefore, having a liver makes you a great baseball player. But it's not true because you have to say how many people with livers are bad at baseball, right? That's the denominator. You don't know how many, 100 baseball players, but what's your denominator? Same thing with cancers. So we got 100 colorectal cancer and we said, wow, all 100 have genetic mutations. Therefore, genetic mutations cause cancer. But you didn't look at the opposite problem. How many normal cells had the same genetic mutations but didn't get cancer? And it turns out that it was a lot. So the answer, and this was from 2002, said that about 4% of the genome could be mutated and you'd still not get cancer. In other words, if the human genome has 25,000 genes, you could get 1,000 gene mutations and still not get a cancer. And in fact, that's what we found. So when they looked at normal cells, so this was from Nature Genetics, 2012, and they basically took people and they looked at their normal cells and they said, how many have cancer, how many are cancer free, and how many mutations are there? As you get older, you get more genetic mutations, that's just normal. And people with cancer tended to have more genetic mutations than those without cancer, so that's fine. But they noted that the vast majority, if not all of the aberrations that were observed in the cancer-affected cohort were also seen in the cancer-free cohort. That is to say that you could have the same mutation in an oncogene or a tumor suppressor gene and still not get cancer. That was not the critical thing. There was something else we were missing. This was from Science in 2018. Again, they're taking normal tissue from people. This was esophageal tissue from biopsies, basically. And they said that, okay, as you get older, what you see is a lot more of these mutations. If you look at sort of people in their 20s, they have very few mutations. And then late in life, in their 70s and 80s, they have over 2,000 mutations per cell. So you're getting a lot of mutations and you're not getting any problems. And the issue, of course, is that for us as clinicians, this progress needs to lead to treatments. And because there was way too many mutations, there wasn't one or two, there was like 70, 100, you didn't have treatments. So therefore, if you look at the survival rates of people with cancer, they were not doing well at all. Like the overall survival from the 70s to the sort of 90s, 2000s was fairly stagnant. It just wasn't making very much progress at all. And this is in stark contrast to the other major killer, the number one killer of people, which is heart disease. So if you look, this is the deaths per 1,000 patients. So you had, in 1950, you had like 586 deaths per 100,000, which dropped like by 2/3 or 3/4, something like that. And strokes, we did really well. Pneumonias, we chopped in about to 1/3. And cancer, well, cancer was about the same. So this was very different than every other disease that we had. So this whole idea that this of somatic mutation theory sort of just didn't work because it didn't lead to good treatments. Yes, there was mutations. For sure, there's lots of mutations, but it turns out there's mutations in normal cells too. But the problem is that cancer's mutations are not random. They're all focused around a very specific number of traits. That is, they grow, they're immortal, they move around, and they use the Warburg effect. It's not random at all. And this idea that they're all genetic clones is completely wrong. There's actually heterogeneity everywhere, but this heterogeneity wasn't random. It was targeted because everything turns out looking the same, right? So if you have a person with colon cancer and they have 1,000 mutations or 100 mutations, and the person sitting next to them, cancer patient B, has another 100 mutations, different ones, they're all targeted to the same thing because clinically they look the same. There's a growth, there's metastasis, there's spread, that kind of thing. And this leads to sort of the next big step from the 2000s to today, which is the evolutionary paradigm of cancer. And the idea is that we're once again trying to go one step deeper into trying to understand what cancer is. That is, we're not saying that there's no genetic mutations. Yes, there are. But what's driving these accumulated genetic mutations that is causing the growth that is causing all of these things? That's the real question, because as we go deeper and deeper, we're understanding more and more. We know the causes of cancer, a lot of the causes of cancer. We're trying to figure out the mechanism. And we know there's genetic mutations, but this idea that they're random was completely not correct. There's something that's behind it. And what is it that's behind it? And this whole paradigm sort of started in the sort of late 2000s when 2007, the National Cancer Institute started to think, wow, we're not getting anywhere. Like, we're starting to see that this somatic mutation theory, this genetic paradigm is going nowhere. So they funded a number of thinkers, including Paul Davies, who's actually not an oncologist. He's an astrophysicist, astrobiologist, actually. And he thinks about life on other worlds and so on. And they said, well, we need different people to think about this cancer problem. And we want you to think about it. Here's some money. Please help us. So he sort of says that, well, he was able to break free of a lot of the sort of biases of his predecessors because he had no background in cancer. So he asked the stupid questions that nobody else wanted to ask, which is, what is cancer and why does it exist? And of course, that is a very good question because cancer is completely baffling. It is not like anything else we know. It comes from our own cells. That is, if somebody develops breast cancer, it develops from their own normal breast cancer. So each cancerous cell sort of develops independently because person A and person B have nothing to do with each other, and yet they develop the same disease. So what is it and why does it exist? The other thing that he noted is that cancer is not a human disease. It's actually as old as life itself. If you go back in evolutionary time, you can see that cancer exists in virtually all multi-celled life forms. So even the Hydra, so this is a picture of Hydra, it's one of these microscopic animals that you study in high school under the microscope. You put a drop of water. They found cancers, they found tumors in these Hydra cells. So even the very most primitive multi-celled animals develop cancer. Not only that, but almost every single cell type in the human body can become cancer. And how does that work? Because a liver cell has nothing much in common with a blood cell or a testicular cell or whatever. So if you're studying human cancers, it's not that useful if you're trying to understand cancer if cancer predates humanity itself. So he went back and started to think about how did life on Earth evolve and where did cancer fit into this whole story? So if we go back to how life on Earth develops, we developed, through a process of evolution. There's prokaryotic cells, which are single-celled organisms and very basic. And then they became eukaryotic cells, which are still single-celled organisms, but much more organized. So you start to see the plasma membrane, the endoplasmic reticulum, and so on. And that was a huge jump forward in terms of complexity because, the nucleus and these specialized Golgi apparatus and stuff, that was a huge step forward. And the next major step was really the jump to multicellularity. So when you consider single-celled organisms compared to multi-celled organisms, there's a massive difference between the two. And the most basic difference is that single-celled organisms are always going to compete with other ones. So you have a bunch of bacteria, for example, they all compete with each other. They will kill each other. That is how fungi developed, penicillin to kill those other single-celled organisms. So they will want to kill each other. They're competing with each other. When they start to aggregate into multi-celled organisms, there was a massive change in the cellular priority because now you're trying to cooperate with other cells. So you organize the cell into bunches of cells, which organize into organs, which organizes into the sort of multi-celled organism. But there's a very big difference because you don't want these cells to go around killing each other. So as we evolved, we had to develop additional instructions that went on top of the original instructions. So the original instructions is sort of survive at all costs, kill everybody, compete. You had to put additional instructions to say, don't kill everybody, don't compete, cooperate. And one of the things that you see the difference between single-celled organisms and multi-celled organisms is that their growth of single-celled organisms is basically uncontrolled. They just grow. So if you look at bacteria in a petri dish, for example, they just grow and grow and grow and grow, and they will keep growing until there's nothing left to eat. That's the only thing that limits them. They won't stop. Whereas a single-celled organism doesn't do that. A multi-celled organism doesn't do that because growth is very, very tightly controlled. You don't want your liver just growing and growing and growing until it shoves your lung, up your nose. It's not what you want to do. You want to grow to a certain point, then you need to stop growing. And it's not limited by resource availability. It's largely independent. It's going to grow no matter what. Even if you eat a lot, your liver is only going to grow to a certain size. It's not going to become the size of a boulder, for example. So single-celled organisms grow. Single-celled organisms are also immortal. So you take a bacteria, you can keep growing that bacteria. And it will keep growing as long as you keep putting stuff in, which is, again, different from the multi-celled organism, which is controlled by the telomeres, of course, and that's the Hayflick limit. So you take a cell, a normal cell, you try and grow it. Each time it reproduces, the telomere shortens, and then you reach this Hayflick limit where you cannot grow anymore. So single cell immortal, multi-cell immortal. We know that single-celled organisms also grow a lot, move a lot. So they have all these different ways, even though they don't have legs, they have all these different ways of getting around because it's to their advantage to be able to move. So a bacteria, for example, even if it can't move, it'll go into a spore and it'll get blown by the wind or it'll get picked up by something else and get moved around because there's always a chance that there's more available nutrients elsewhere. They know that if they stay in the same area, they're just going to lose all their resources. Whereas multi-celled organisms have multiple ways of sticking them in place. So you want these adhesion molecules to stick all the cells so that your liver doesn't move around. And the fourth major difference between single-celled organisms and multi-celled organisms is that single-celled organisms use fermentation. That is, they take glucose and they produce lactic acid. Whereas multi-celled organisms use oxidative phosphorylation. And this is, of course, the Warburg effect. So if you look at the comparison of cells within a multi-celled organism compared to single-celled organisms and compare them to cancer cells, what you can see is that cancer cells and single-celled organisms are practically identical. That is, the priority for this cancer cell is the cancerous cell itself. It's every man for himself, just like the single-celled organisms. Whereas for the multi-celled organisms, your liver, for example, will die off if it needs to. because it doesn't want to take over or take over resources. Everything is geared towards survival of the organism, not survival of the cell. But cancer cells are not like that. They want to compete, same as the single cell, whereas the other one wants to cooperate. These are the sort of hallmarks of cancer: growth, immortality, movement, and glycolysis. You can see that there's a stark difference between the multi-celled organisms, where they don't do this, and single-celled organisms and cancers that do this. So cancer cells actually behave very much like single-celled organisms. But you can actually go much deeper. There's actually a number more ways that they are alike. Despecialization, so cancer cells start to be able to grow themselves and do all kinds of other things. They're autonomous, they do their own thing. They have host destruction, so just like a bacteria will destroy the tissue around it, so will cancer cells. You see this exponential growth, so cancer cells will just keep growing and growing. It will invade into new territories, whereas normal cells will never do that. So if you have a liver cell, it doesn't invade through the lung sort of thing, whereas cancer liver cells, of course, do that. They'll compete with resources, and they have genomic instability. That is, they will mutate and they will change, just like single-celled organisms. If you think about it, the disease of cancer and the infectious diseases are very, very similar. That is, in no other conditions do you face the same problem with resistance. So single-celled organisms, of course, you give antibiotics, they develop resistance. You don't get that, for example, with a kidney stone. You blast it, it doesn't start resisting it. You'll get new kidney stones, but you don't get the same kidney stone all of a sudden resistant to that. It just doesn't happen. Whereas cancer does the same thing. You treat it with radiation or you treat it with chemotherapy. When it comes back, it's now resistant to that treatment. You can't use that same treatment again. We don't have that in virtually any other diseases, because these are the same thing. These cancer cells and these single-celled organisms behave exactly the same way. They invade tissue, right? You don't, if you have kidney disease, it's not like it starts invading into the pancreas. They metastasize. You have metastatic infections and metastatic cancers. You don't get metastatic kidney stones. You get the resistance, you get genetic mutations, the cells evolve. So in infections, of course, you get new, the evolution and the resistance. And cancer cells evolve too. So if you take somebody who's been treated several times and you check their genome against the original cancer, it's completely different. And they have secretions. That is, they will secrete things and get them. So the question is, how did these two diseases, why are they so similar? Like, how did it get to that stage? And the only thing sort of powerful enough in biology to do this is evolution. So this is the sort of idea that came through, is this behind this evolutionary paradigm, is that you started out, all the cells within our body started out originally with a unicellular genome, because we all evolved from single-celled organisms. As we became multi-celled organisms, we added all these genes, like for immortality, right? We want to be mortal. We want to limit growth. We want to limit movement. We want to do the more. So, but you didn't get rid of the original programming. You had all the original playbook underneath, and you just added stuff on top so that you could get, hey, stop growing. Hey, stop living so long. But the underlying sort of core was still always there. And what the idea is that the kernel of cancer exists in every single one of our cells. And as you expose these cells to damage, so you damage the area that's controlling the growth, all of a sudden you have unlimited growth. You damage the area that's controlling immortality, all of a sudden you get immortality. So that is the kernel of cancer, this unicellular ancestor, is already preformed in every single cell of our body, and really of every single cell in all multi-celled animals. And that explains a huge amount of the behavior of cancer. That is, how does cancer exist in every animal, multi-celled animal that we know? Because that's how we evolved. That's from evolution. How is it that every single type of cell in our body can become cancerous? Well, it's because we all have that kernel of cancer that comes in. It explains how these cancer cells can grow and metastasize and develop resistance. And it explains how cancers look the same. So this is a very interesting thing because you can take a patient with breast cancer in 1920 in the United States and compare it with somebody who lives in Asia or Africa in 2020. So 100 years apart, different races, different continents. And yet the breast cancer will look exactly the same, but they've evolved completely independently of each other. How is that possible? How is it that they look the same? And it's because it's this sort of preformed kernel of cancer that is now expressing itself through this genetic damage. And it's as we take out these sort of additional pieces that we expose this. So this is not a forward evolution. It's actually an evolution backwards in time. That is, we're not getting more evolved, we're going backwards into a more primitive state, which is in fact exactly how the pathologists describe cancer. They're primitive cells. They're de-differentiated cells. Anaplastic, the word means back, ana means back, and places means formation. It's all describing a backwards evolution towards a more primitive state. And that's why we all look alike, which is called an atavism. It's an evolution backwards, not to become more evolved, to become less evolved, to become more survivable, because that's the sort of original survivable sort of wild animal ancestor that we have. And the idea is that as we sort of fall backwards into this unicellular state, lung tissue and liver tissue, for example, start to look more and more alike because they're more similar. And that's why how they can grow in each other's environments much more easily. And so it really explains a huge amount. The idea is that so cancer cells, they behave exactly like single-celled organisms. It explains why genetic mutations are not random, but they should target primarily the state from unicellular genes to multicellular genes, because the origin of the cancer really lies in that jump from single-celled organism to multicellularity. So can we find evidence that that is actually what's happening here? So in fact, this is exactly what they're starting to find. So if you look at the unicellular genes, and for most of history we're single-celled animals, and then you have this sort of multi-celled gene, this overlap period is where you find cancer. So this is from 2017, and they looked at all the mutations of cancer. Instead of saying, well, which gene is mutated, what they did was they asked the question is, these genes, are they newer genes or older genes? So they divided them into 16 phylostrata, which is to say that here's our most ancient genes, unicellular genes. And these are our more recent genes, evolutionarily speaking. These are the more recent genes, where not which genes are mutated, but from what era are they mutated. And what you see, of course, is that they're all coming in from the unicellular portion, this part where it jumps to multicellularity. All the sort of recent genes, that's not where you're finding all the damage. You're finding all the damage, all the mutations, just in this transition. This is the same thing. These are the genes in millions of years. And these are all human genes, overmutated and undermutated genes. And you see they mostly fall in line with what you expect, except right here. The most ancient genes are the ones that are targeted in cancer development. It's not that, it's not all the recent stuff, it's the old stuff because it goes way back. And again, this is the same things showing sort of all cancer genes. Where is it that you find the mutations? And there's two spots sort of right here, very, very ancient genes, the most ancient genes, and right here, which is this transition from sort of single-celled organisms to multicelled organisms. Right there is where you find the gene mutations. Same thing here, 2016 from another Nature Communications. So again, most genes that are multicellular, they sort of fall within where you expect. But this part right here is where you find all the genes of cancer. Right here, from when you're going from a single-celled protozoan to multi-celled organisms all the way up to humans. So it's really describing an evolutionary process in this transition state. And the question is why? Why is it reverting to this unicellular state? It's an extraordinarily complex thing to go from a sort of multi-celled organism back to a single-celled organism. So why is it going to do that? And the answer really is evolution. It's not just randomly developing into cancer, it's evolving back towards its sort of ancestor. And it requires three things. One is genetic heterogeneity, which we know it has. It requires time. So it's a chronic process. And it requires a selection pressure, right? Just like this is the Darwin's finches. So he said, well, you know, the environment says that, hey, if there's a lot of nuts, you get these thicker beaks because that's what's favored. The birds where there's a lot of fruit get these thinner beaks. So it's the selection pressure. What is the selection pressure in this case? And it's probably sublethal injury. So if you look at chronicity, what's important, of course, is that if it's an evolutionary process, it's going to take time. So acute injuries are not going to do it, but chronic injuries are going to do it because it's going to give it the time it needs. So you can look at hepatitis A, for example, and hepatitis E. Severe liver disease, but acute, not chronic, and doesn't cause cancer. Hepatitis B and C, absolutely. You get acute gastritis, doesn't cause cancer, but you get chronic gastritis, all of a sudden you get cancer. Same thing with inflammatory colitis versus a chronic ulcerative colitis. You get a burn, you don't get that much cancer, but chronic sun exposure, like sunburns, you get cancer. You know, radiation was the same. So acute radiation, they worried about Hiroshima, the bomb, when people were exposed to massive levels of radiation. And they worried that people would get cancer. Turns out they didn't really. This was a study of the Hiroshima and Nagasaki survivor studies. And this is the sort of excess amount of cancer that they saw. So the blue is how much they expected to see. The white is how much they actually saw. So you see maybe 5% extra, but you're talking about massive levels of radiation exposure. But the key was that it was a single acute event. It was not a chronic event. And that's why you didn't see the cancer, which was a great thing in that case. But you needed to be chronic. And the single sort of overriding hallmark of these organisms is survival, right? It's survival. It's sort of like the survivalist in the woods, right? He's got to be self-sufficient. He's moving around. He's, always trying to grow his land. He's going to compete with other people trying to invade on him. That's what the unicellular organism is. It's like the survivalist, right? And it's focused on its own survival, not the organism's survival. So the idea is that the selection pressure doesn't come from sort of minimal injury, because if you have a little bit of injury, you're just going to repair it. If you have massive injury, the cell is just going to die. But it's in this sort of in-between state where you have chronic sublethal cellular injury, where you get this breakdown like of law and order, you get this anarchy. And the cells have to adapt because they've been injured. So if they don't adapt, they're going to die, and they don't want to die. So how are they going to become more survivable? Well, they're going to start competing with each other, and they're going to start shedding off these more recent sort of instructions to cooperate with each other, and they're going to sort of look out for themselves. That is, they're going to revert back to that single-celled organism mindset. And they do this by getting rid of all of those excess baggage. That is the underlying sort of playbook of the single-celled organism is now coming out. And it's just like anarchy. Whenever you have a breakdown of law and order, there's looting, every man for himself, every woman for himself. You start to, form bands and kill each other and that kind of thing. So it's the exact same thing, but it needs to be chronic, and it's this sublethal injury. And that really explains a lot. So as you evolve into multicellularity, what you see is that the tumor is this damage to these other things, the survivalist core sort of comes out because the handbook that was there from the unicellular organism already is there and just waiting to come out. It's not a random genetic process, but it's a targeted evolutionary change back towards single-celled life. And the interesting thing about it is that we already know that cancers undergo an evolutionary process. So this is an evolutionary tree. This is called branch chain evolution. And you see, bacteria branch off here and, animals and fungi branch off here. We can do exactly the same thing with cancers. So they've done this. This is from the New England Journal of Medicine, 2012. They took a bunch of sites. They sampled this M1 tumor, M2 tumor, and they sampled all these metastases. And they basically did exactly the same thing. You can follow exactly how this tumor evolved. So you go from a normal tissue, and then you get these mutations, then you split off and you get these mutations, these mutations, these mutations. And this is how you get all of these different heterogenetic things. But what's driving the mutation in the first place is this sort of trying to become increasingly survivable. So in this case, the way we approach medicine, the cancer, is that we have to say, well, what is this? It's chronic sublethal cellular damage. The cellular response is an evolution back towards this sort of survivalist single-celled organisms, and therefore all sources of chronic sublethal cell injury are now potentially carcinogenic, which is actually something we already knew. So these processes are not necessarily mutagenic. That is, they don't necessarily cause genetic mutations. Some of them do for sure, but all you need is the chronic sublethal cellular injury to activate this evolutionary process. So even normal things like Barrett's esophagus is normal stomach acid, but in the wrong place, it causes this chronic subcellular injury. But the stomach acid itself is not mutagenic. Same thing with inflammatory bowel disease. There's nothing mutagenic. It's not like ionizing radiation. It's chronic inflammation. Anything that causes chronic inflammatory disease, any type of cirrhosis. So even fatty liver disease, which is, excessive fat in the liver, there's nothing mutagenic about the whole thing. And yet, any cirrhosis is going to increase the risk of hepatocellular carcinoma because it's this chronic sublethal damage. That's what's happening. And over time, so there's the chronicity, it's activating evolutionary processes. Sunburns, the same thing. Radiation. So the exact same thing that we treat cancers with is carcinogenic because you're causing chronic damage, chronic inflammation, chronic damage. So the same thing with chemotherapy. The very drug that you're using to treat cancer causes cancer. And we've of course known that forever. These are called secondary tumors. You get that with radiation, you get that with chemotherapies. We know very well. Same thing with transplant medications. Some of them definitely lead to that. So this is the idea. This is this evolutionary paradigm that we've now moved towards, which really explains a lot of the things that the somatic mutation theory never could. That is, all of these questions like, why do all these things that don't cause gene damage cause cancer? Why does cancers look the same? Why do you have so many cancers? Why do all animals get cancer? Why do all cells get cancer? So none of that could be explained. But as we move towards this sort of chronic sublethal cellular damage, which really all of them do, so infections do the same thing, chronic infections, and chemicals and smoke and all of them, they cause this reversion to unicellular existence because that's our sort of evolutionary ancestor, which is going to cause targeted genetic mutations because it's an evolutionary process towards cancer, which is just that expression of the single-celled organisms, which then manifests as the hallmarks of cancer. That is, these things grow, they're immortal, they move around, and they use the Warburg effect. So that's the answer that we're coming up with, is that all of them are genetically diverse, but they look identical at the pathological level. That is, two breast cancers look the same under the microscope, but when you do the genetics, you can see that they're completely different from each other. And then why does it exist? Well, it exists because that's our ancestor. That kernel of cancer has always been there. Every cell can become cancer. Every animal can get cancer. And the cancer is a common, not a rare disease, which is actually a very interesting notion, because if you think about the number of mutations that it takes to develop, a cancerous cell, if it was a random process, it would never happen. It would happen like 1 in a billion or 1 in 10 billion people would get cancer. Because if you're trying to develop a new tumor that has all these abilities to survive while the body tries to kill it, it could never do so. If it was really the somatic mutation theory, we should have virtually no cancer. Instead, it's one of the most common diseases that we face. So that's, again, one of the key things that this evolutionary paradigm can really explain that the other ones can't. And there's a huge number of therapeutic implications of this, some of which we use already. Well, there's therapeutic resistance. That is, you can't use the same drug over and over. There's combination chemotherapy. So you think about how you treat a disease such as tuberculosis. You don't use one anti-tuberculous agent, you use 4 or more. Same thing with chemotherapy. Our success rate when we went from one chemotherapy to these regimens of chemotherapy improved tremendously. So once again, you see, cancer is very much like an invasive species, like a new invasive species. That's exactly how we treat it. Screening, should we screen as much as we do? Because sometimes it's never going to develop. As long as you get rid of the underlying conditions, that is the sort of chronic sublethal damage, it may never develop. Do you need maximally tolerated dose? So this is, of course, the sort of paradigm of chemotherapy that we've labored under for 50 years, which is give as much drug as possible without killing the patient. That was the idea behind a lot of the autologous stem cell transplants and so on. But it turns out that you might not need that. So just like with infections, you don't necessarily treat an infection with 2 months of maximum dose antibiotics. Sometimes you just need enough to get through. Does the cancer need to be eradicated or just controlled? Because it's not, it is a natural process there. And of course leads to the next sort of big wave in cancer therapeutics, which is now if we look at the paradigms, we have the growth paradigm. So we're going to kill, cancer is a cell of growth, so we're going to kill cells. Then you had the genetic paradigm, which is that we're not going to try and kill cells. We're going to try and fix the genetic problem. And it succeeded initially, but then fell apart. Now we get to the next one. That is, we are thinking now about cancer as an invasive species. It's a new species that has developed due to evolutionary processes, but it's basically an invasive species to us. And you might think, oh, that's ridiculous. How can you evolve a separate species? Well, that's how our body sees it. Because we have natural killer cells that kill foreign cells. And they see those cancer cells as foreign cells. So now as you move into the evolutionary paradigm, you have to think these are actually completely new organisms, new species of cells. Well, we have ways of dealing with that. That's our immune system. And that's where immunotherapy is now this big, huge wave. I mean, we've seen a lot of successes already, but it's very complicated, of course, because immune treatments are just, there's a lot going on there. So we've already seen a couple of instances where we've had these great immunotherapies, but that's the next big wave, because focusing in on those genetic treatments, I mean, if you think about it, Gleevec imatinib was developed in the 90s. It was a revolution. It was great. But over the next 30 years, if you think about the number of drugs that came out of that genetic paradigm that have really shifted the disease like imatinib did, it's like probably less than 5. Like you could probably count them on one hand. That's after 30 years of research. Like, yeah, you had Herceptin, you had a few of them, but it's not a lot. And there's no future in that, because if you develop 5 drugs in 30 years, you're never going to move the needle in terms of cancer. You need to start to think about it in this new manner, this evolutionary paradigm, which is just explains a lot more. But that's really only the beginning. It actually has huge implications for the progression of cancer and the metastasis of cancer, because the way you have to think about metastasis, for example, is completely different if you're thinking about it from an evolutionary standpoint. I cover this if you're interested in my book, The Cancer Code, and talking about some of the recent research about all that. Thanks very much.
Thank you. That was quite a lot of food for thought. It's interesting. I guess if people have questions, again, just use the raise hand function. When programming last month was Parkinson's Awareness Month, and during that talk, we were made aware that melanoma is increased in patients with Parkinson's. And just happened to come across an article that says that there might be a link between those two conditions because of disturbances in the microbiome, which is kind of another way of looking at these diseases in general, that cancer and neurodegenerative conditions might have a common factor to them in the microbiome and disturbances in that. And I'm just going to throw a question, because the way you presented this is like it's as if bacteria are all individualistic and atavistic, in a sense. But I like to think of the microbiome as an organ. And I don't know who's supporting who, whether it's the microbiome supports our bodies but they work cooperatively. And what's interesting to me also in recent literature is that tumors, there's a new research into tumors having their own microbiome, which is kind of, I think, a radical idea. So I just want to throw those ideas out there, and I guess you can comment if you like.
Yeah, and I think that these sort of paradigms really lend themselves to new ways of thinking about it, because the somatic mutation theory was such a dead end and really just dominated cancer so much, to such a degree, even though the cracks were already appearing, those Vogelstein papers, and he's a huge, person in cancer research. I mean, that was from almost 10 years ago that he already showed that there's no way this sort of somatic mutation theory is possibly correct. So it sort of opens up the door to saying, hey, how is the microbiome affected? Because this is a huge thing. If we're talking about chronic inflammation, if we're talking about nutrition, whether we're talking about all of these things, it makes a big difference because this is a much more inclusive sort of model. For example, the link between immune suppression and cancer. We know for sure if you suppress the immune system, like transplant, you get cancer. Unquestionable. But the somatic mutation theory just cannot explain that at all. Why would immune suppression lead to that, right? You have immune surveillance, you have all that, but why would that lead? It's not mutagenic. Like immune suppression is not mutagenic. So it opens the door. So the link with Parkinson's, I'm not sure where the link is, but at least now you can start to think broader as opposed to thinking purely as, hey, how is this going to damage the genome? How does this cause gene mutations? Because it was such a narrow sort of focus. So I think that this is actually a very important advance, and sort of, and nobody's trying to say, oh, we're all wrong. In fact, we were right. It's just that we're taking the next sort of step in progression. And when you're trying to think about treatments and stuff, now you can sort of think about all these other things that cause or might cause cancer.
Okay, great. Okay, so it looks like we've got a few questions coming up. So Leonard, you're number 1. So just wanted to unmute yourself and identify yourself. Maybe describe what kind of doctor you are and where you're coming from, and then ask your question.
I'm Leonard Sadinsky from North York. I'm retired. I'm thinking along your lines, Dr. Pupko. Should we not be blurring the distinction between things like infections like HIV, hepatitis B and C, and what we call cancer? Should we still have a separation there, or are we blurring? Should we be blurring that line?
Well, I mean, the infections are a little bit different. Like hepatitis B and C cause cancer, of course, and there are specific agents that are going to treat hepatitis C, for example. But it's the sort of reaction to that chronic infection or chronic inflammation or chronic damage that is leading to the change. So one is the change agent, and this is sort of the reaction to it. As the sort of cells are trying to survive, they sort of lose that normal cellular law and order, and they become sort of anarchists, and they kind of go by themselves. So there's definitely a blurring of the lines here, as opposed to thinking of it as just, hey, you have an infection, hey, it causes a gene mutation, and this is all gene mutation, so let's affect this gene mutation. So there's definitely a blurring of the lines, and I think that the sort of now that when I look at it, it's like, wow, the similarities between cancers and infections is sort of really radical. It's like, wow, this is just a crazy sort of idea, but there's a lot of data to suggest it. So yeah, absolutely. I think that the blurring of the lines is actually great in terms of spurring new thinking into a field that has unfortunately had very little success compared to other things, because we're treating it according to a paradigm of gene mutations, right? The somatic mutation theory. So we're out looking for the last 30 years, we're out there looking for gene mutations, but we should have been looking for why these genes were mutating. So the immunotherapy and all that, compared to say cancer, where we've made way better progress, strokes where we've made way better progress, right? So of course, hopefully this spurs the next sort of wave of big innovations.
Thank you for your superb presentation. I really appreciate it.
Thank you.
Just thinking now as an idea came to me, so it's not well developed, but that's okay. This is what the program's all about. But in your book, you mentioned how circulating tumor cells come home. And I'm just thinking in broader terms, in that maybe cancer cells are just looking for home and they never find it. So they just come back home to join the former, where they started out from. Like the same way there's migration in nature, that, the salmon always come back to where they were born. Maybe they're trying to find the microbiome, but there's something in the way. They're just trying to go home. That's like all of us. I remember when I was a kid and I had surgery I remember the only thing I remember from the surgery is like me saying, I want to go home. I want to go home. So maybe
I actually think you're totally right. That's actually what I talk about in later parts. There wasn't enough time in this one, of course. But the idea is that, the old idea is that metastasis is a late event. So that the tumor gets big, and chunks break off, and they metastasize. That's not what happens at all. So what likely happens is that very, very early in the course of this developing tumor, before you can even detect it, it's already sending metastases out. So say you have a colon cancer. Before you even know it's there, it's sending out metastases. Now, these metastases are identified as foreign cells and get slaughtered. They all die. And maybe once, as it develops over and over, they die. But waves of cancer cells just keep coming out. Eventually, one makes it through the bloodstream, and then it lands on, say, in the lung. But it's a hostile environment. It's a liver cell, doesn't know how to live in the lung. But once in a while, it comes back to the original site. And as it's gone through, this has become sort of a more survivable form of that cell. So what came out was, this sort of, idealistic young cell. It comes out, it's been harried, it's been, through the gulag of the bloodstream, it comes back, it's a hardened cell, and it's more survivable. Now that competes against the original cell, and now you get new strains developing within that original tumor site. So what happens is then you're going to have several implications of that. One is that metastasis is detected early, and that's exactly what you find. So you can now detect circulating tumor cells, CTCs, long before you see any metastases, because the sensitivity of our assays are so much better now than 30 years ago that we actually know that there's all these circulating cancer cells all the time. They last like an hour or less in the blood before they're wiped out by the immune system. And that's why when you suppress the immune system, you get cancer, because they can no longer perform this immune surveillance. And so that's a fascinating thing. But as you get these waves and waves and waves of recolonization of the original site, you're going to get new subpopulations of tumors, which is exactly what you see. When you have a lung cancer, you don't have one homogeneous mass of cancer cells. You actually have multiple subpopulations with completely different genetics in all of those sites. And that's only going to happen if you have this sort of waves of sort of cancer cells going out, then coming back, then repopulating, cancer going out, coming back, repopulating. And each time it's just an iterative process. It gets a little better, a little better, a little better, and then keeps growing, growing, growing. But it's a totally different thing, because if metastasis is early, now you can do what a lot of people are trying to do, which is liquid biopsies. Take some blood, look for these circulating tumor cells, and then trying to identify your cancer even before you can possibly see it, which is, again, super fascinating and comes completely out of this evolutionary paradigm.
So I would argue that maybe you kind of presenting the cancer cells as these rogue cells that are just all in it for themselves. But in a sense, they're coming home to their family as the mature survivors and creating a new social system. And that's why there's so many different types of cancer cells within a tumor.
Yeah.
So
because each time it evolves, it's a little.
Yeah, but they're social creatures in a way.
In a way.
Organizing themselves, right? For the
Yeah.
Great Revolution, whatever it's going to be. But
Yeah.
there was a theory that I read about years ago, and I think it was, I don't want to attribute it to the wrong person, so I won't even mention his name. But he said something very interesting, and I haven't seen it repeated. And you did a lot of research for this book, so maybe you can answer this question. A lot of the times when people have their tumor removed, it's a single isolated tumor, and then it's removed, and boom, suddenly all these mets appear. And the theory was that the primary tumor was producing chemicals to suppress the metastatic tumors. And once you remove the primary, it's actually detrimental to do that. And possibly you should be treating with chemotherapy beforehand. I'm just wondering if you came across that, or if there's an oncologist in the house who can comment on that, or a surgeon who removes tumors and then wonders if that was such a great idea or not. Any thoughts about that?
Well, it explains it perfectly, and you also, the sort of evolutionary paradigm sort of explains exactly how this could happen, which should never happen according to the sort of somatic mutation theory. Same thing with late metastases and stuff. For example, there's one case of a young lady who had a melanoma when she was 20, then died of a car accident at 50. They took out the lung, transplanted to this recipient. The recipient developed melanoma. It's like, what? Where did this melanoma come from? Well, when they sequenced it, it came from the donor. So it had lain in the lung for 30 years, just completely dormant, because it was sort of holed up there. It couldn't get out because it would get killed in the blood, but it wasn't, that it was strong enough to sort of hold on. Then when it gets into the recipient, the lung transplant recipient, the immune system is suppressed and all of a sudden it just blossoms. Within a year, I think that patient died. So same sort of thing. There's something else going on here that this sort of idea of, let's look at it like an invasive species. That is going to be a much more successful. That's going to explain a lot of what we see in metastases, in the progression of cancer and so on. And a lot of these diseases, for example, when we talk about over-screening, which happened, for example, in South Korea. They screened all these people, they found all these thyroid nodules. The rate of thyroid cancer went up tenfold, but the number of deaths from thyroid cancer didn't budge at all, because you're just finding a whole lot of sort of indolent tumors that would be controlled by your people's own immune system and would never have been a problem, which is exactly the same thing in breast cancer, right? This sort of overdiagnosis of DCIS and so on. And, a number of bodies have actually started to say, we shouldn't be doing mammograms in young people. And that's this idea that, hey, a lot of these are never going to get worse because they're just kept in check by the body. It's an invasive species, yes, but you don't need to treat all of them. So you wind up risking sort of over treatment of this. And also explains a lot of other aspects, like how does obesity lead to cancer? We know there's 14 types of obesity associated cancers. Well, obesity is not mutagenic. It has nothing to do with somatic mutations. So how does it lead to cancer? Well, as you look at it as a sort of invasive species, you say, well, obesity is going to, increase nutrition, increase insulin, which is a growth factor. Cells like the glucose, cells like the growth factors. Now you can piece it all together and say, hey, this is the link between obesity and cancer, because this whole gene mutation thing, being obese doesn't mean you have more gene mutations. It just has nothing to do with that. So this idea that it's much more than a genetic disease is very powerful and explains a lot of like what you're talking about, these phenomenon that people could never explain.
Which is also an interesting thing is spontaneous regressions.
Yeah.
I was reading in the literature a couple of cases of renal cancer that spontaneously regressed after getting COVID. Not that anybody's in favor of COVID, but it was kind of fascinating to see that two people actually benefited in the world. So
well, the spontaneous regression, that was the idea between Coley's toxins, which is an old form of immunotherapy. So they had this guy one time who had sort of inoperable cancer. Then he, in the old days, in the 1800s, he laid next to somebody with erysipelas. He got infected. The inflammation from that infection actually stimulated the immune system and wound up completely reversing his cancer. His cancer went away completely. And this is the same thing. The stimulation of the immune system wound up stimulating it, and it's like, hey, there's all this cancer sitting here. And so it started attacking it. And that was the idea behind Coley's toxins, which is an old form of immunotherapy, where you would actually inoculate bacteria. It's a fascinating story. People would inject this bacteria into the tumor. This was from the 50s or something like that.
we still use it today, like BCG for bladder cancer.
Exactly like BCG. So you're injecting toxins, like bacterial toxins, into the tumor to promote inflammation, which is going to stimulate the immune system. It's going to now say, hey, there's all this cancer there, and it's going to get rid of it. And BCG is the sort of like first line treatment. Well, how does it work? Well, you wouldn't understand that if you didn't understand the evolutionary paradigm. It's a totally fascinating area.
Okay. So moving on, somebody is called iPad2. Maybe you could open your camera and also identify who you are and where you are as a physician, and ask your question.
Yeah, so my name is Dr. Genevieve Gabra. Jason knows me very well. Hi, Jason. So Jen here. So I have a question for you about obesity, because I work in obesity medicine. I used to work with Jason in Toronto Metabolic Clinic, and I work in Scarborough Health Network as an internist as well. My question is more about the intermittent fasting. Is there a link with intermittent fasting and reversing or decreasing risk of cancer? I don't know if it's a long shot. And my second question is metformin use. I see that metformin is used a lot for cancer patients like melanomas and So what's the link with metformin as well with cancer? So my first question with intermittent fasting and reversing hyperinsulinemia, probably decreasing inflammation, and I don't know if it's prevent cancer or delays it. And my second question is the metformin.
Yeah, so that's questions. And the thing about fasting is that there's not a lot of data. So there's a little bit of data from its combined use with chemotherapy. So people have done some studies where they fast for 24 hours before chemotherapy, then they get the chemotherapy, then they eat, and it seems to enhance this sort of tumor effect. And the idea is that when you are fasting, you're going to put your cells into a more quiescent state. So you know that a lot of chemotherapy targets rapidly dividing cells. So as you fast, your cells are going to become more quiescent. So you're going to be relatively protected against the side effects of the chemotherapy, whereas the cancer cells can't do that. They're always grow, grow, grow. So therefore, they're going to get exposed to the full effect of that chemotherapy, which is targeting sort of cell cycle. So that's about the only data. Most of the, can fasting prevent cancer probably works its way through obesity. So we know there's obesity associated cancers. So therefore fasting allows you to lose weight. Presumably you're also going to reduce the risk of cancer. So breast cancer, colorectal cancer, they're all obesity associated cancers. Hyperinsulinemia is also a well-described risk factor. So again, insulin is a growth factor. So anything that encourages things to grow are going to be sort of linked to cancer. So insulin is sort of a prime one. So there's a lot of interesting data around fasting and how it can help, but there's not a lot of good data, unfortunately. But as a treatment for sort of metabolic disease, we know that type 2 diabetes is associated with cancer. And we also know that the obesity is associated. So it's going to treat both of those. The question about metformin is very topical because it was also used for breast cancer. And it actually affects a different mechanism through the AMPK, which is a different nutrient sensor. So nutrient sensors are mostly like insulin. It basically tells your body, hey, nutrients are coming in, and those nutrient sensors also are growth factors. So when you eat, insulin goes up, it tells your body food is available, and also tells your cells to grow, because that's how they're so tightly linked. In evolutionary times, insulin was not a sort of nutrient sensor. It's actually used as a growth hormone. So we use it for both. And metformin affects this other one called AMPK, which looks at sort of total cellular energy. And there was some excitement a little while back that it could actually reduce breast cancer. But actually, I think just this week in JAMA, they published a study that metformin, in fact, didn't have any effect on breast cancer. So there's some observational stuff about how it could be useful, but metformin, unfortunately for breast cancer anyway, just didn't work out, because probably it's a less important factor than insulin and mTOR.
Okay, Jason, your camera seems to be off. Okay, Margaret McPherson, you have a question?
Yes, I do. Thank you. Can you hear me?
Yes, we can. Can't see you if you want to.
I'm trying to start the video, but I can't.
Okay.
Dr. Fung, I'm a retired dermatologist. Dr. Fung, first of all, thank you for an absolutely fascinating talk. It's very interesting. My question is apropos of what you said about people starting to do liquid biopsies, looking for cancer cells floating around in the blood. And I wonder if that would have, if you think it would have, any applicability to the earlier diagnosis of pancreatic cancer. And if so, do you have any idea how soon it might become clinically applicable?
I don't know. The thing about these circulating tumor cells is that they have a very, very short half-life. So people are trying to develop them because there's so much promise in them. But, the sensitivity of us being able to detect tumor cells has gone up so much that we can detect sort of like, there's probably like less than a few hundred cells by the time you get the blood, and it's not going to last very long. So you have to get to it right away. So I don't know the specifics, but I do know that there's a huge amount of excitement because you can actually measure these circulating tumor cells before that pancreatic cancer is, you can see it on CT scan, right? So it's so, there's so much promise there. And if you can develop a panel, for example, then you can take a tube of blood and screen for all the cancers. It could be pancreatic, it could be lung, it could be breast, it could be everything, right? So the sort of idea that you could do that is, the promise is so huge. So could it happen? I don't know. I sure hope so. I mean, it would be an absolutely interesting, and huge jump forward in therapeutics for us. But how soon that's going to be, I just don't know. Right now, it's just a pipe dream, but we know we can detect them. Can you do it in a clinically meaningful way? I don't know, unfortunately. I know there is like several companies sort of pursuing it because, of course, there's always some biotechs. The pot of gold at the end of that rainbow is huge.
Thank you very much.
Thank you.
So I'm just wondering what kind of reception this idea is having within the oncology community. I know when you wrote your book about diabetes, you upset quite a few endocrinologists. Are you getting any feedback from our colleagues in the cancer field?
I haven't heard a lot, and I think it's because I mean, a lot of this data is sort of a little bit, one, it's new, but a lot of people are talking about it already. Like a lot of the top sort of oncologists are talking about it. They don't present it exactly like this, but they understand that there are these evolutionary sort of tendencies. So even though it's, it was very controversial a few years ago, but I think as things move forward, people are starting to accept that this is sort of an evolutionary disease, which is why it's a different disease than almost every disease that we see. We just don't have evolutionary diseases that we're treating. We're trying to come up with completely new ways of treating this disease because it is not a disease that is like any other. So that's, but I haven't heard too much. And most of it is, it doesn't affect therapeutics that much. So it's not as controversial a topic. That is, I'm not trying to say you shouldn't listen to your oncologist, right? I'm saying that you should still do whatever they tell you to do. But it's just an interesting, from a scientific standpoint about thinking about this disease, which has so far eluded us as to what it is, right? That's the fascinating part. Nobody talks about this like at all. So it's like you go to these cancer, meetings or anything, like who's talking about what is cancer? Like nobody. Like people are talking about, oh, this gene and that gene and this drug and that drug. There's endless talks about that, but no talks about sort of the more interesting from a, intellectual standpoint. What is cancer? Like, what is this disease? How did it develop? Why did it develop? Like, these are just sort of philosophical questions for the most part that don't affect treatment quickly, but are important to understand things like metastasis, like circulating tumor cells, like, maximally tolerated therapies and all that sort of stuff. But it doesn't change treatment itself. So I don't think it's as controversial. To me, it's more of an interesting story, but the progression is interesting. This idea about why cancers use the Warburg effect. I mean, again, super interesting. Doesn't change a lot to me, but a very, very interesting discussion about why should cancers use this Warburg effect. Right?
Well, it's interesting because that's why I invited Dr. Reiter. Unfortunately, it looks like unless Russ is out there, I think he's left. But here's a person who's devoted his entire life to studying melatonin and its therapeutic effects, and it's got, and he was promoting. He has written. So Russell Reiter is one of the world's leading experts in melatonin, and he was talking about how melatonin has an anti-Warburg effect, so it's working against one of the mechanisms of cancer, and yet people aren't, it's a simple intervention to initiate, but it's not taken up. I even spoke to one of my cancer patients a couple of months ago and said, okay, well, why not introduce melatonin to the mix. And the oncologist was against it. It's relatively benign, but pretty powerful stuff, theoretically. I mean, there aren't any studies on it. It's kind of, so there's a lot out there that's theoretical that could have great clinical implications, but we just don't seem to.
Well, yeah, because it doesn't fit into the right paradigm, right? If you're looking at it from a genetic paradigm, you say, why should I care, right? Why? Like this whole Warburg thing is, to me, again, very, very interesting, because what you have is a cancer cell that is using glucose much less efficiently than with oxidative phosphorylation. So, you're making 2 ATP plus 2 lactic acid versus 36 ATP. Why would you do that? Especially since you need energy to grow. And there's a few reasons, but one of them, which I think Russ had alluded to, was that the lactic acid may actually benefit the cancer in a number of ways, because essentially what you're doing, the cancer cell, is it's dumping all this acid out, which is protecting itself from all the other normal cells, sort of creating its own little home, that's toxic to all the regular cells, because all this lactic acid sitting around. You get all this inflammation. As you get inflammation, and this is called tumor-associated inflammation, you get white cells that come in, which will release growth factors, because that's what inflammation does, right? You get a cut, you release growth factors to try and heal it over. So you've got growth factors coming in. This lactic acid is actually highly advantageous to the cancer cell. So therefore, if you look at it from that standpoint, you say, this is an invasive species. It's using the Warburg effect to produce lactic acid. Lactic acid is what it wants to do, because if you kill other people, you get an advantage. So you dump acid all around you, you get protection. And therefore, melatonin would say, okay, well, let's cut off this sort of supply of lactic acid, and maybe that's going to improve survival. So it makes perfect sense from an evolutionary standpoint why melatonin should work. But you'd never understand that if you're stuck on this sort of, hey, we're just going to, if you're in the growth period, we're just killing cells. We're just, cutting, burning, and poisoning cells. That's what we do. Like, that's 2 paradigms ago. Like you've got the growth paradigm, you've got the sort of the genetic paradigm, and now you've got the evolutionary paradigm, which explains all of those things.
Yeah, so back to it's the idea of the microbiome, and the I was reading interesting that that in certain chemotherapies, when the chemotherapy is ineffective, by changing the microbiome, you can actually make the chemotherapy more effective. So definitely, the cancer cells are acting almost like bacterial cells, and maybe communicating with their friends in the gut or elsewhere. Or
again, it
even
opens
closer.
up that possibility, because if it's acting like an invasive species, then the microbiome may have a role, because they interact with each other, right? So maybe it may become more friendly or less friendly, or the other bacteria may. But it opens up that entire possibility that never existed before.
Yeah. Okay. I think people are thinking really hard about your presentation, but I don't see any more hands up. So there's Vivian, you want to ask a question?
Thank you, Vivian Ramby. I'm a cardiologist. Thank you for this novel and revolutionary approach. But I would like to suggest taking this to paradigm 2.0. The use of the term evolution means a self-organization. And the Nobel Prize for Physics last year was one for chaos and complexity in complex things like climate science and other crises, climate change. Similarly, there's a self-organization in chaos theory, which is like evolution with determinism, what appears random, but is not random. And that's the Nobel Prize for Physics. So similarly, there could be a self-organization that appears random, but not random, that comes out of the complex dynamic interactions, and a bit like heart disease and a heart attack with a sudden plaque rupture. and again, using the term evolution, maybe the process is like evolution, but it's chaos and complexity. And there's some people are now saying even evolution of species is partly chaos and complexity. It is not adaptation, etc. It appears random, but it's chaos. And there are many books out now about genomic chaos and complexity, etc., as a cause and development of science. So I agree with what you're saying, but the term evolution, maybe that's chaos and complexity, and the emergence of uncertainty, etc., in this complex system.
Yeah, I mean, I think that's an interesting idea. I mean, and certainly it may be. I mean, the evolutionary paradigm is one that the oncologists use, actually. So I think that it's actually, it's possible that it's all chaos now, but you do still have to explain why all of the mutations in cancer fall in those very sort of specific, age of genes that is the kind of, as we evolve from single to multi-celled organisms, that's where all the genes become mutated. So there's a few issues there. Like if it's true chaos, then you shouldn't see that. You should see, you should see mutations across all age genes, right? So, but I agree with your point. The point is actually sort of a broader one. That is, we have to move past what I think probably you and I both learned, which is this sort of somatic mutation theory of like, one hit, you affect one oncogene, then you affect the next oncogene, and then you affect the tumor suppressor system, and then boom, you get cancer, right? That's what I learned anyway, and it's just not true. So getting beyond that, sort of towards other ways of thinking about it, whether it's chaos that looks, random but isn't random, or whether it's true evolutionary, or it's activism, which is this sort of movement backwards in time. It's really all about going past the sort of the gene based theory, so that we can develop new treatment. So I'm open to that for sure. And of course, there's going to be new theories. There's going to be things that there's going to be another paradigm that comes down that's going to be more inclusive. But the point is that it's all a sort of this layering of knowledge, right? We started with growth, then we say, why is it growing, right? Then you get the genes, and they say, why are the genes changing like that? Then you get sort of evolution, but maybe it's chaos theory, and I'm totally open to that. So thanks. That's a great point.
Yeah, I think maybe again, thinking out loud here. In a sense, this is what we do as human beings. When we're stressed, we revert to our primitive self, right? We revert to fight or flight mode, so that, and then we can restore back to a stable, secure self. So in some ways, maybe these cancer cells are reverting to their primitive mode as a temporary measure. So they look chaotic, but they're basically adapting to the injury until they can heal and move back to being a regular cell. But there's a block there that keeps them from reverting back to where they came from. So, I think we're all, as humans, we're all, if you look at ourselves, we're units, we're cells, we're made out of cells, but we're just one cell, we're one unit, right? And I think there's a lot, the way our cells behave is the way a lot of us behave. So we can revert to primitive behaviors, but we can also rise to quite high levels of being human. And so we can be civilized, or we could be what is going on, unfortunately, a lot in the world today, the opposite. Okay, Vivian, do you have any other points? Because Vivian gave us an entire talk on chaos and complexity, so I'm sure he can go on all night. But did you have any other points that you wanted to make? Because your hand is still up. So.
Oh, sorry, my hand should not have been up. I should have taken it down. But since you asked me, I'll make one quick comment.
Okay.
So Jess, if you use the term invasive species and regressing to different cells, this sort of gives an impression of a purpose of something happening with a design. However, not just chaos theory, but complexity, which comes after chaos, talks about a self-organization that's uncertain, and maybe what you call invasive species is the process of the self-organization, creating patterns. So there are different patterns in different places that are self-similar. Again, from chaos theory fractals, they develop roughly similar but different depending on their original circumstances. So maybe the process is that of chaos and complexity. But where everything else you say, and the data and the original cells and the data and so on may be valid, but the process is chaos complexity.
It's certainly possible. And I think that's a fascinating, I don't know enough about complexity or chaos theory I actually hadn't seen any of that in any of the stuff I looked up, but certainly it sort of made sense to me. I mean, there could be another process that's going on that's not truly an evolutionary process, right? Evolution is a very specific process of sort of natural selection. So you have time, you have genetic heterogeneity, and you have a selection pressure. So you're adapting to a specific selection pressure. And what you're saying is that it might not be a selection pressure. It may be, you know, chaos instead, and this sort of self-organization. So it's possible. I mean, I like that idea.
Add a quick comment right on that. There's a Professor Heng at Chicago and Harvard who talks about genomic chaos. So at a chromosome level, there's chaos that causes things to happen as opposed to random. And that can lead to cancer. Now, just as an aside, in terms of the history of this thing, I was invited in the year 2000 to Cambridge University to Rethinking Cancer think tank, because I knew a bit of chaos, but the person who was organizing it had just written a paper on breast cancer as chaos. So this has gone back many years, and there are many books out now talking about it. So I think it's something we should think about.
Yeah, no, I agree. And I think the more we think about sort of outside the box of where we had sort of put ourselves, which is genes, and more genes, because we spent a lot of time looking for all these genes. It took like 20 years for the Pan-Cancer Atlas to come out, right? We spent a lot of money looking at genes. If it's really chaos theory and genomic chaos, that's a totally different thing. Like you would approach it from a completely different angle. And that's why I think it's important to think about these sort of different things. This is the way things are moving sort of since the sort of 2010s towards this sort of evolutionary thing, but it's not sort of crossed over to most physicians. They haven't thought about it much outside of that, which is why I thought it was such an interesting topic to write about.
Okay, thanks, Vivian. Martha Wright.
Oh, hi. I'm a psychiatrist here in Toronto. I'm interested in pursuing, coming back perhaps to your point, Dr. Pupko, about in terms of on a higher level human behavior of becoming, our better selves versus becoming more our primitive selves that we like to refer to in psychiatry. But but coming back to looking at on a cellular level, if the process that we're talking about for cancer is an evolutionary process of returning to a more simplistic single cell organism driven mechanism, then the one question I was having is, well, what is there that we could be implementing to try to restore, because the genes to those evolutionary genes are still within the cancer cells, presumably, if they haven't totally mutated, that you could revive an activation towards becoming more organized, moving back towards more of a multicellular organism. So that is, we're talking about immune therapy, but immune therapy, I'm assuming, may just immobilize the cell or kill it. Is that, or is there a way of somehow encouraging the cell to reestablish those previously dominant programs of organization and, anti-migration, et cetera, because so I'm just wondering if there's an overriding mechanism people are thinking about to try to revert the cell to a more of a multicellular functioning organism.
Yeah, that's a great question. And it mostly comes down to the environment, because if you're thinking about it from an evolutionary terms, then what's driving the gene mutation is the selection pressure, which is the environment. That is, if you smoke, then you're creating an environment that is going to sort of stress the cell. So then when you stop smoking, of course, you change that environment, which is driving this sort of move towards survivability of this single cell. And that's going to, and we know that's going to lower your rate of lung cancer in the future. So you really have to look at what's sort of driving that whole process. So for example, obesity associated cancers, then it's like, well, what is it about the what's happening in the obese individual or type 2 diabetic individual? And you might say, well, perhaps it's hyperinsulinemia. Then you can say, okay, well, let's get them to lose weight or bariatric surgery or medications, that kind of thing. Because what you have to do is trying to change that environment, because that's what's driving the gene changes towards survivability. So it's not easy to do, of course, but once you identify it, then you can do it. So sort of like, and that moves you beyond, say, screening, which is different, right? You're just trying to pick it up early and then cut it out, sort of thing, say like colonoscopies and mammograms and stuff. So you're trying to pick up these sort of early sort of environmental things, which we should be doing anyway, like stopping smoking and treating hepatitis C and getting rid of, fatty liver as a cause of cirrhosis, those kind of things, sunburns. So all of those things that we're trying to do are all environmental things that are affecting that. And that's the way that you affect it, because evolution is a response to a changing environment. So it's the environment that's changing. We need to change it back.
I guess, but that's a sort of just interrupt here for a minute, but that's kind of a preventative of identifying the risk,
right?
And you starting to see the cellular changes, perhaps, or the environment changes.
Yeah, no, you're right.
In a frank cancer environment,
in a frank cancer,
we could just remove as much of it you can, and then hope. Like I'm thinking of picking up the remaining cells, which is where everything is being left at. How do you somehow manage those remaining cells, because they're the ones that become troublesome?
Yeah, and the other example, maybe in an established cancer, is the idea that you don't need maximally tolerated dose, or MTD, which has been used in certain studies for, say, prostate cancer. So instead of treating, sort of giving as much drug as you possibly can to a prostate cancer patient, because the drugs actually cause cancer too. The radiation causes cancer and the drugs cause cancer. Instead, just trying to contain it. And once you get above a certain level, then you give more drug, so there are certain applications, although it is harder to think about. But again, probably people would have to think about it a little bit more and do the experiments. But MTD is one of those issues where I could see that affecting actual treatment of a cancer. So not just whacking it. You know how we think about cancer: hit it as hard up front as you possibly can. From an evolutionary standpoint, that may or may not be the best thing to do. So that's one area where you could change potential treatment.
Yeah, and I would again encourage you to go to PubMed and look at cancer. Just do the search terms cancer and melatonin. You should have I think you should go to bed early tonight, but next night, we don't want too much of this blue light coming at us to throw off our melatonin levels. But the principle there is what you're talking about is helping the cells to revert back to normal, right? So you can make it's an example. By no means am I saying melatonin is going to cure cancer. That's far from what I'm saying. But it's an interesting starting point to see that It's a possibility, and as physicians, we underutilize it, and it's hard to make recommendations because there aren't any clinical studies to back up the theoretical considerations. But it's kind of fascinating to look at that literature. But bottom line is that cancer is still a mystery, and I'd like to thank Jason for giving us some pieces of the puzzle so we can try to solve it or try to understand it the best we can in our own minds based on what we know to this day. I think you did an excellent job of reviewing the history of treatment and the history of the ideas. And I guess one last question for you, Jason. What's your next book? And when will you coming back to the lounge to share with us?
I actually didn't even mean to rewrite the cancer code. I just found the story of the whole sort of evolutionary paradigm just so fascinating. And I just thought, oh, okay, I'll write about it. But
Well,
I don't have anything I'm working on right now. I'm sort of working with more clinical stuff, trying to sort of help people with diabetes and so on.
All right, back to your day job. Okay.
Yeah.
Well, thank you so much for a great evening and keeping with the mandate of the Doctor's Lounge to think outside the box, and then you've done a great job. So thank you all for joining us. Next session is actually pretty soon. It's June the 15th. We'll be looking at another mystery, fathers. So please come in and join us. Thank you.
Thank you.
Okay, good night.